Hi, welcome to the 5th lecture in my series on the coronavirus. This lecture will be divided into 2 parts. In the first part, I'm going to talk about how antibodies can be effective therapies. How exactly do they work to stem the progression of a viral infection? In the second part, I'm going to compare and contrast 2 approaches for treating the virus that invoke the immune system. So first, vaccination. Second, passive immunization. All right, so with that, why is it that antibodies are effective therapeutics? So recall that we had the structure of the coronavirus, and I'll just, draw quickly here. We have the membrane envelope on the outside, we have the RNA on the inside, the nucleocapsid protein, and then we have different proteins out in the membrane. So we have the E protein, the M protein, and the spike protein. And the spike protein can exist in different conformational states, and I've already described that. So, this is the initial state, and then it goes through some changes where it undergoes cleavage event, and you have the spike here that would destabilize the membrane that's going to drive fusion of the virus with a target cell, either a target cell plasma membrane or endosomal membrane. So these are just 2 different configurations of the spike protein. So when you raise an antibody, remember I said antibodies are Y-shaped molecules, so they look like this. For simplicity, I'm not going to draw the different sites at the tips of the Ys that enable the specific contacts. I'll just draw the antibodies as Y molecules. So, when you have an immunological response to a virus, the immune system, it's not like it's intelligent and it knows beforehand exactly what antibodies to make. It just makes any possible antibodies that it can against any viral antigen that it can see. And so you may end up with many, many different antibodies to the virus, and I'll just draw a few in here. So, you could have some that bind in that part or this part of the M protein, antibodies that bind to the E protein in different parts. For example, antibodies that bind to different parts of the spike protein on the side here, maybe in this linker region, maybe in this region right here, maybe in this region right here, and so forth. Okay, so many, many different antibodies. You can literally have potentially hundreds or thousands of different antibodies that form against the different proteins of the coronavirus. And that would even include proteins on the inside, like the nucleocapsid protein. Now, some of these antibodies might not do anything, right? They might bind the virus, but they might not have any effect on the function of the viral proteins. So maybe antibodies like these might be of that sort. Even some of the ones that bind the spike protein, like, these antibodies might not really do anything to the function of the spike protein. However, antibodies that, bind here and here could potentially block the function of the spike protein, because if you recall, the tip of this piece here is required to destabilize the membrane that the virus is going to fuse with, and an antibody that binds there could potentially interfere with that function. Likewise, this part of the spike protein has to bind to the ACE2 receptor on a target cell, and an antibody binding that same surface could compete for that for access to that surface with the ACE2 protein, and thereby could potentially block viral infection. So that's one way in which antibodies can work, is that by binding on the virus, they can directly interfere with the function of viral proteins in a way that prevents the virus from getting into cells, all right? The other way in which antibodies can work, so that's the simplest way. So that's just direct inhibition. Another way in which antibodies can work is that once the virus is coated with antibodies, and remember, there's 60 copies of the spike protein. So if you make a ton of antibodies against the spike protein, potentially you have 60 antibodies bound on the surface of the virus. And in fact, if antibodies bind different parts of the spike protein, you could imagine you might start having hundreds of antibodies literally coating this virus. Well, there are cells in the innate immune system that their job is to run around looking for things that are coated with antibodies, because if something is coated with an antibody, then it's probably foreign, okay? And those substances that are coated with antibodies are eliminated by a process known as phagocytosis. And what this means is literally cell eating. So you have a particular type of specialized cell known as a macrophage. And a macrophage is in some ways similar to a dendritic cell, where what it does is it vacuums stuff up from the environment. However, the difference is the primary function of a dendritic cell is to process those antigens and then upon activation, go to a lymph node and activate lymphocytes in the lymph node to essentially to educate the adaptive immune system. On the other hand, the chief function of a macrophage is just to get rid of stuff, that's foreign in the environment. So it's a vacuum like the dendritic cell. It runs around vacuuming stuff up, and then it just destroys it, right? It sends it to the internal Cuisinart for shredding. And so if you have many antibodies, whether or not they're inhibitory, if they're coating your virus, that can trigger phagocytosis of the coated virus by macrophages and then destruction of those viral particles. So those are the chief way in which antibodies can help provide an antiviral effect. In the next lecture, I'm going to cover how passive immunization with antibodies differs from vaccination.